So, in the last video, we reviewed the major components of action potential and what causes them. So, based on that, you should be somewhat familiar with a diagram that looks like this. What we see here is a diagrammatic representation of an action potential. And as we remembered, resting potential was established by sodium-potassium pump generating a slightly negative charge inside the neuron in relation to the outside of the cell. This was negative 70 millivolts. And once triggered by a stimulus, the sodium channels opened up, and the sodium rushing into the cell resulted in the inside of the neuron depolarizing, approaching zero and then overshooting it a little bit. But this could not be sustained for long, and then the repolarization takes place as sodium channels close and potassium channels open, resulting in the potassium leaving the cell. This re-establishes the negative charge inside the neuron and actually undershoots the value of negative 70 millivolts a little. And this undershoot was important because it gave us the period of hyperpolarization, which we knew as the refractory period. During this time, no other action potential can typically be passed, so it is a little recovery time for us, preventing messages getting confused. And that was our diagram for the action potential. But you might have asked that how do we actually know that this is the case? How can we measure the action potential? And this is actually something that I used to do. The method is rather simple yet very effective. We use a fine-tipped microelectrode which gets put inside the neuron, while another ground electrode is located outside the neuron. And then it's just using a voltmeter to record the values. And this is what it really looks like in a lab. Manipulating this fine-tipped microelectrode is sometimes a real pain, but when it works like in a textbook. It's quite remarkable, really. The other thing that I want to highlight as a bit of a review is the role of myelin around action. In terms of the speed of the electrical impulse that is passed, here we see a non-myelinated axon. So we have no myelin around it. And therefore, the conduction is slow since the action potential has to pass step by step throughout the entire length of an axon of a nerve cell. And the explanation for why this is so slow is that the fact that it takes time for ions and channel proteins to move across the membrane, and this was needed for each step of the progress of the action potential. This is a little bit different now. Here we have a myelinated axon. So we have these fat tissue sheds around the action like beads of pearls necklace, if you wish. And what myelin does here is that it enables a process where action potential has to be created only in the gaps between the individual myelin blobs. Therefore, it is like the action potential could jump from one gap to another, and therefore the potential travels faster down the axon. I hope that this clarifies the role of a myelin a little bit. Remember that in addition to speeding up the propagation of an action potential, it also provides support and insulates the structure too. The last thing that we have to review relates to the process that we can see an electron microscopic figure here. And this is, of course, the synapses. So a junction including the small gap between where the presynaptic action ends at the axon terminal and the postsynaptic neuron starts. So remember that two nerve cells were not physically connected to each other, but there was a small gap between these. And this gap was known as a synapses. And it was here where we needed to use chemicals, known as neurotransmitters, to pass messages across this gap. In the name of being precise, I should mention that there are also electrical synapses, but we will focus now on the chemical ones. So these chemical synapses are much more common than the electric ones. And there is a real science behind neurotransmitters, how they are made, released, pass the space, and then bind to the receptors on the opposite side, and even how extra neurotransmitters are taken away or neutralized. But this is not something that we will be able to go in a great detail here. There is a field called neuroscience, which I minored in university, that one focuses on all these questions, among other topics. And I think that this is a rather nice diagram that shows how the action potential arrives to the action terminal and causes the voltage-gated calcium channels, remember these from our video of action potential, to open. As a result, calcium now enters into the axon terminal. And this calcium entering into the axon terminal causes the synaptic vesicles, small membrane sacs containing neurotransmitters, to release their content into the synaptic gap. And now this neurotransmitter passes across the gap, eventually binding into the specialized receptors that are its target on the opposite side of the synapses. This in turn causes ion channels on the post-synaptic membrane to open and now initiates an action potential in the second cell. Finally, as everything that needs to be done is done, the neurotransmitter's effect on the receptor is terminated and it is either taken back to the original action terminal, deactivated by an enzyme that breaks it down, or it may diffuse away from this site. So that is a nice explanation of how chemical synapses work. And your textbook will talk a little more in detail about different neurotransmitters and summation of the action potential signals and so on. Please review this information from the textbook. But for now, this is all I wanted to highlight on this video. I will see you in the next chapter.